In the animal kingdom, certain creatures exhibit the awe-inspiring ability to regrow lost limbs, a phenomenon that has captivated scientists for decades. Among these remarkable beings are lizards and the amphibian known as the axolotl. Lizards, particularly the green anole, can regenerate their tails in a matter of weeks, while axolotls can even regenerate limbs and vital organs. The axolotl, often referred to as the Mexican walking fish, has become a focal point in regeneration research due to its extraordinary capabilities. Unlike most animals, the axolotl does not form scar tissue over its wounds, allowing it to regenerate tissues perfectly. This includes not only limbs, but also parts of its heart, spinal cord, and even portions of its brain. Understanding how these creatures accomplish such feats offers a tantalizing glimpse into what might one day be possible for humans. Scientists are eager to unravel the genetic and cellular pathways that enable such regeneration, hoping to apply these findings to human medicine. The implications of such discoveries could revolutionize the way we approach healing and recovery. As researchers delve deeper, the potential for humans to one day harness similar regenerative abilities becomes more than just a dream. Tissue regeneration is a complex process that involves the replacement of damaged or lost tissues with new growth. In animals like lizards and axolotls, this process is highly efficient and well coordinated. The key lies in their ability to initiate and control cellular processes that lead to the formation of new, functional tissues. Central to this process is the formation of a structure known as the blastema. When a lizard loses its tail or an axolotl loses a limb, cells at the site of the injury undergo dedifferentiation. This means they revert to a more primitive stem-like state, which allows them to proliferate and form the blastema. The blastema serves as a reservoir of undifferentiated cells that will eventually differentiate to form new tissues. This process is regulated by a series of signaling pathways and genetic networks that ensure the correct cells are produced in the right quantities and at the correct time. Proteins and growth factors play crucial roles in signaling these pathways, guiding the cells through the various stages of regeneration. By deciphering these mechanisms, researchers hope to unlock the potential for inducing similar regenerative processes in human tissues. Stem cells are the unsung heroes of the regeneration story. These remarkable cells possess the dual abilities of self-renewal and differentiation, making them indispensable for the regeneration of tissues. In regenerating animals, stem cells are mobilized to the site of injury, where they give rise to various cell types needed to rebuild the lost structure. For instance, in the axolotl, stem cells are responsible for recreating the bones, muscles, nerves, and skin of a regrown limb. This ability is facilitated by the cell's ability to receive and respond to biochemical cues that direct their development into specific cell types. The potential application of stem cells in human medicine is immense. Scientists are exploring ways to harness the power of stem cells to regenerate human tissues and organs. Techniques such as induced pluripotent stem cell technology allow for the generation of stem cells from adult tissues, which could then be directed to form new tissues. This approach holds promise for treating a wide range of conditions, from injuries to degenerative diseases. Advances in understanding stem cell biology continue to push the boundaries of what might be possible in regenerative medicine. The quest to unlock the secrets of regeneration has led scientists to a variety of animal models, each offering unique insights. The green anole lizard and axolotl are two such models, but researchers are also exploring other species like the reindeer. Reindeer, known for regenerating their antlers annually, provide a different perspective on the regeneration process, particularly concerning bone growth and vascularization. Recent studies have shed light on the molecular mechanisms that guide regeneration. For example, scientists have identified specific genes and chromatin dynamics that regulate the regeneration process in axolotls. The use of advanced technologies such as CRISPR gene editing and single-cell RNA sequencing has revolutionized regeneration research. These tools allow researchers to dissect the genetic networks involved in regeneration with unprecedented precision. Insights gained from these studies are not only advancing our understanding of regeneration, but also informing strategies for manipulating these processes in humans. By understanding how these proteins work, scientists hope to find ways to reactivate similar pathways in adult tissues, paving the way for potential regenerative therapies. The notion of humans regrowing limbs, much like lizards or axolotls, might seem like science fiction. Yet the scientific community is actively exploring this possibility. The goal is to enable humans to regenerate lost tissues and limbs, offering hope for those with traumatic injuries. Promising research focuses on dormant regenerative pathways in humans. Some believe humans have latent regenerative capabilities silenced over time. Understanding genetic mechanisms could reactivate these pathways. Technologies like stem cell therapy and tissue engineering are key. This could lead to breakthroughs in limb and tissue regeneration. The dream of regrowing limbs is becoming a tangible goal. Despite the exciting prospects, the path to achieving human regeneration is fraught with challenges. One of the primary obstacles is the complexity of human tissues and the intricate coordination required for successful regeneration. Unlike the relatively simple tissues of some regenerating animals, human limbs involve multiple tissue types including bones, muscles, nerves, and blood vessels, each requiring precise integration. Another significant challenge is the body's natural tendency to form scar tissue rather than regenerating the original tissue. Scar formation is the body's rapid response to injury, providing a quick fix to close wounds and prevent infection. However, this process often precludes the possibility of full regeneration. Understanding and potentially modulating the scar formation process is a critical area of research. The ethical considerations surrounding regenerative medicine also pose challenges. Manipulating human tissues and the potential for genetic modification raise questions about safety, consent, and the long-term implications of such interventions. Continued funding and collaboration across scientific disciplines are essential to overcoming these hurdles and making human regeneration a reality. As with any groundbreaking scientific endeavor, the pursuit of human limb regeneration raises important ethical questions and societal implications. One of the primary concerns is the potential for unintended consequences. The manipulation of genes and cells to induce regeneration could have unforeseen effects, both at the individual level and within the broader ecosystem of human health. The concept of equitable access to regenerative therapies is another critical issue. As these technologies develop, ensuring that they are accessible to all segments of society is paramount. This includes addressing potential disparities in healthcare systems that might limit access to advanced treatments for certain populations, exacerbating existing inequalities. The ability to regrow limbs could redefine our understanding of disability and rehabilitation. It could also impact cultural and social perceptions of physical integrity and identity, prompting a reevaluation of what it means to live with or without certain physical attributes. The ethical discourse surrounding regenerative medicine is essential in guiding the development and application of these technologies. As the field progresses, ongoing dialogue and engagement with diverse stakeholders will be crucial. Looking ahead, the field of regenerative medicine holds immense potential for transforming healthcare. As our understanding of regeneration advances, so too does the possibility of developing therapies that can repair and restore tissues damaged by injury, disease, or aging. These advancements could revolutionize the treatment of a wide range of conditions, from traumatic injuries to chronic degenerative diseases. One of the most exciting prospects is the development of regenerative therapies that can be tailored to individual patients. Personalized medicine approaches, which take into account a person's genetic makeup and specific medical needs, could optimize the effectiveness of regenerative treatments. This could lead to more precise and efficient healing processes, minimizing complications and improving outcomes. Additionally, advancements in bioengineering and nanotechnology are paving the way for novel regenerative strategies. These technologies enable the creation of scaffolds and biomaterials that can support tissue growth and regeneration, enhancing the body's natural healing processes. The future of regenerative medicine is bright, with each discovery and technological breakthrough bringing us closer to a new era of medicine. The study of limb regeneration in animals like lizards and axolotls provides a fascinating window into the future of human medicine. Scientists have uncovered mechanisms that allow these creatures to regrow lost parts, offering insights that could one day be applied to humans. The progress made is a testament to scientific inquiry and innovation. Human limb regeneration is a beacon of hope for those facing limb loss. As researchers unravel regeneration complexities, novel treatments grow nearer. These advancements could transform lives and reshape medical care. Ethical considerations and equitable access must guide us. The journey continues, fueled by hope and the promise of human regeneration.